Ready?
All right, well, I think we can go ahead and get started. So thank you all for coming here today for Rebecca Salomon's defense. I mean, it's quite a huge crowd. I normally draw much thinner crowds. I don't know what that says about me, but it clearly says a lot about Rebecca. So, well, I guess it's kind of almost an end of a journey of 5 years. And I guess it started really even before that 5 years. Rebecca really contacted me about 6 months before she even started grad school, which was like I just started my lab. Someone's contacting me that they're joining grad school in 6 months and they're interested in what I'm doing. It was like, okay, cool. That should be my first student then. And indeed, she eventually did join the lab, as you can guess. And one thing she was interested in from the get-go is one of the projects we're working on, which is respect to cardiac innervation. Now, one thing that we did find previously during my postdoc is that nerves are quite important in regeneration, but we really had no full understanding of how, the heart is innervated during development, more importantly, in response to injury and regeneration. So we kind of wanted to go on a bold project and try to figure all that out. So it was really a kind of a daunting task, but Rebecca quite relentlessly developed her own tools. And I guess that was one limitation for that project, but she really powered through a lot of techniques that you will hear a lot about today, that she pretty much set up on her own in the lab that really set the stage for her to answer these questions. And as you'll see, she made a quite remarkable findings that I think will change our understanding of how, nerves function, not only under homeostasis, but in response to injury and disease, and potentially a novel therapeutic avenue in response to injury. So Rebecca's, I think, is now a first author on 3 papers. 2 are pretty much done, and 1 is going to be resubmitted maybe later today, I think. Well, we'll see about that. But she's done quite a remarkable job, really, over the past 5 years, and she's been involved in other projects with respect to, what we do in the lab and in terms of development and regeneration. Not only that, but she was funded by her American Heart Association Personal Award, and I can list on many of her speaker awards throughout meetings and posters. That's even including no meetings during the pandemic. So she clearly had a lot of those afterwards. Well, I want to stop here because I'm sure you're all here, not for me. So just want to, I guess, give the mic back to Rebecca to take you through her wholehearted journey over the past 5 years. So, Rebecca.
Thank you. All right. Let's dim some lights. Well, I first want to say a huge thank you all for being here in person or by Zoom. This project was not done alone. It was done with a huge amount of support personally, supporting me as a person and supporting me as a scientist. I will get plenty of time at the end to acknowledge everyone. That's really helped me throughout my journey, but I just want to say a massive thank you to you all for your support, your kindness, and I am extremely excited that today I get to share with you my thesis work. It's titled A Wholehearted Journey, Mapping Patterns and Pathways of Cardiac Disease, Development, and Regeneration. And so to give you a bit of an outline of what I'm going to get to touch on today, I'm going to go into a background, a little bit of an introduction, get to share with you some of the methods that I developed for my project, go into the main bulk of my thesis work, which really focused on looking at the cardiac nervous system in a new perspective. And then just for a bit, share some future directions. So let's dive on in, starting with some introduction. I'm going to be talking a lot about the nervous system. Your nervous system is extremely complex. It's allowing you to understand and interpret the world around you. It's allowing you to take in your senses, your sights, and bring those signals into your brain, distributing those to all of your organs. I would argue that the nervous system is the most complex element of your body. It's connecting all of your organs. It's regulating your life, your thoughts, your movements, your actions, your voice, everything that you do on a daily basis. Really, it's made up by your nervous system. And we can think of our nervous system, as an extremely complex process, but I can also share with you an example that, makes it a little bit easier to interpret. Well, let's say you're listening to something really fun. Well, I know one thing that we're all listening to today. Everyone's very aware that today is the release of Speak Now, Taylor's version. So I know what I was jamming to this morning, and we're all loving it, right? Yeah. Okay. So Speak Now, Taylor's version, we're hearing those lyrics. They're getting transmitted directly into our brain, where we can interpret her lyrics and also decode those Easter eggs. And when we understand those lyrics, we're just going to get so pumped. And that's going to increase our heart rate. And that's just one example of how dynamic your nervous system is, and really just allowing you to enjoy your life, understand the world around you, form your thoughts. And we can break down this complex process into really three main parts. You're having a sensory input, in that sense, the music. It's traveling through sensory nerves, getting integrated in your brain. And then the brain is passing along those signals to your organs. In this case, I highlighted the heart, and those are happening through motor nerves. Now, I'm going to be talking a lot about nerves today. So I'll go ahead and break down the main parts of a nerve. You have your nerve soma and your nerve axon. The nerves function much like a telephone. The soma is like a mini brain or a receiver of signals. And so you can think of that as the physical telephone. The axon is allowing those signals to travel extremely long distances, just like a telephone cord might. The body does not have Wi-Fi, it does need cords. And so you have axons that can extend from anywhere from being a millimeter to a full meter in length. They're allowing for the transmission of signals across extremely long areas of your body. And I'll be talking a lot about these nerve axons today. And this is your entire nervous system throughout your body, but each organ also has its own nervous system. And of course, I'm very focused on the heart. So when we think about the cardiac nervous system, we automatically think about how the nerves are able to regulate heart rate and contractility, or the strength of the beating. And it's because we have this direct readout. We know nerves influence heart rate. It's a very clear association that we know a lot about how this functions. So we have a nerve, and it connects within this small region of nodal cells. Nodal cells are also nicknamed the pacemaker cells of the heart because they set the pace for the heart rate. And the nodal cells are the sinoatrial and atrioventricular nodes. Now, these are localized, as you see, in just one section of the heart. So your heart has four main chambers: your right atria, right ventricle, and left ventricle. These fill with blood, allow for pumping. And you see that really the focus of this heart rate is just within one region, the right atria. And so it's within this right atria that the nerves will signal for the initial contraction. It will tell those pacemaker cells to first contract. That will lead to the signals being passed between the nodal cells. The nodal cells will then lead to the atrial contraction. Then it passes the signal yet again to the Purkinje fibers, leading to the ventricular contraction. So atrial contraction, ventricular contraction. And this rhythm is generating your heart rate. Now, your heart rate's not always beating at the same pace, of course. It's changing dynamically. And that's thanks to you having two flavors of nerves. You have your parasympathetic nervous system, which is nicknamed the rest or digest nerves. These are active when you're sleeping, you just woke up, you haven't had your coffee, you've got your parasympathetic nervous system active. But then on a Friday morning, you get to wake up and go to a lecture on the cardiac nervous system, and you're so pumped that your sympathetic nerves start going, your fight or flight nerves. And I hope to, get your sympathetic nerves going today with some of my data. And so it's this dynamic exchange between your parasympathetic and your sympathetic nervous system that's allowing for the balance of heart rate. Now, I'm very interested in these nervous systems, not because of what's known about them, but of course, what's not known about them or less well known about them. And so I've shown you that these nerve axons stretch within the atria and they control heart rate, but they don't stop there. The sympathetic nervous system also stretches within the ventricles. And this becomes a very curious finding, because if we know that the nerves are really known to regulate heart rate and contraction, the atria, well, what are they doing in the ventricles? And this question really remained unanswered in the field because it was thought that the ventricles don't have very many nerves, so What are they doing in the ventricles? Who cares? They're barely there. They're not influencing it. But over the past few years, we've been able to see that that is absolutely not the case. If we look at the sympathetic nervous system, we see there's been some really remarkable findings just in recent years. We see that these nerves are regulating the cardiomyocyte size in the heart. Cardiomyocytes are your main contractile cells of the heart. They compose most of your heart tissue. So the cardiomyocytes are what are allowing for your heart to beat. And they're able to regulate the size of these cells, these cardiomyocytes. They're also able to regulate the rate at which they proliferate during development or the rate at which they divide. And also, most importantly, during homeostasis, their continued nerve signal is needed for the regulation of just the maintenance of cardiomyocytes. So we see that there's this crosstalk where the nerves are talking to the cardiomyocytes are also talking back to the nerves, and that led to the definition of a neurocardiac junction for the first time in the heart in recent years, in 2018 and 2022. Now, that's your sympathetic nervous system, but what about your parasympathetic nervous system? Well, it's quite a different story when we think about your parasympathetic nervous system, because it's thought that the parasympathetic nerves have minimal to no contribution to the ventricles of the heart. And this has really stood in the field for quite some time. Even today, if you go up to a clinical cardiologist and ask them how many parasympathetic nerves are in your ventricles, they'd probably say somewhere between none to a few. But if we look into the field, we can see some evidence that maybe this isn't the case. Maybe the parasympathetic nerves influence a bit more than we're giving them credit for, because we see that the ventricular cardiomyocytes can express a receptor that would allow them to respond to parasympathetic nerves. Well, that's curious that they would express something that they wouldn't need. Also, we see a direct protective effect where the stimulation of the parasympathetic nerves can protect against arrhythmias. Again, an interesting finding if they're not in the ventricles. And lastly, and most importantly, really serving, as Ahmed mentioned, as a catalyst for this project, was the realization and the discovery that parasympathetic nerves directly regulate cardiomyocyte proliferation and regeneration in the ventricles after injury. Now, that was the most striking finding for us and really served as a catalyst for asking the question, well, where are the parasympathetic nerves in the heart and what do they look like? But before I could answer that question, we quickly realized I needed a new way to look at the nervous system. And that's where my chapter 2 begins. I needed to develop a method that I could accurately look at the nervous system. And I really wanted to model it in 3D and intact. I didn't want to manipulate it in any type of way. I wanted to get the most accurate view of the nervous system. And when we think about how we've been looking at the nervous system throughout the years, it's through two-dimensional sectioning. What that means is you're taking a tissue and you're chopping it into very thin slices and looking at each individual slice. Here I have shown you the entire cardiac nervous system in 2D. It is very hard to interpret. You are seeing it correctly. We are taking these nerve axons and we are slicing them. These nerve axons are long, thin strands. As you are slicing them, you are damaging the nerves. And as you're trying to reconstruct the nervous system, you're losing an entire perspective of the proper nerve architecture and a lot of their cell-cell interactions. And it becomes very hard to interpret where these axons are at in the tissue. And so I wanted to take a new approach looking at the cardiac nervous system in 3 dimensions. And when I take that same view, we were quite struck by what we were able to see. We were able to see a dense network of cardiac axons covering the cardiac ventricles more than had been appreciated before in previous literature. And really, there's very few people in the field that will recognize how many nerve axons are truly in the heart. And we're hoping to change that perspective. So I wasn't the first one to ask this question of what do the nerve axons look like in the heart, but I did it in a new way. And so I want to highlight here how people have been asking this question through the decades. And so if we're using the most advanced techniques at the time, we can see here now a timeline of looking at nerve axons and the kind of the development of the field. And so in the 1960s, we're limited to bright field microscopy. This is the use of light and dyes to trace structures. We see this dense black structure, that's a nerve. Looks a lot different than the one I showed you. And it's because you're getting a very limited scope into the nervous system with just some dyes and light. Now, then we have electron microscopy again in the later 60s, and we're able to see some really nice subcellular structures, but those long, nice axons that I showed you, nowhere to be found. It's not really compatible with this type of microscopy. It wasn't until 2010 and we started getting some evidence that, you know, there are some serious nerve axons likely in the ventricles, but we're getting that, again, two-dimensional view. So here in green is a nerve, and then in red is that muscle tissue, the cardiomyocytes. And we see, okay, there's some little nerve axons there, but again, we have no idea of its actual composition in 3D. And it wasn't until 2014, whole mount immunofluorescence, meaning we were able to keep more tissue intact. And we're able to use really powerful lasers to start looking at an intact nervous system. And so we see now, again, the green being the nerve axons, we can see nerve axons, but what we can appreciate is the fact that the red signal is inferenced due to high background. And that means that that's all pretty inaccurate. So we're having to inference quite a bit of signal. We're not quite getting the resolution we'd like. And it was in 2017 where the processes of whole mount clearing and light chain microscopy started giving us an intact view of the cardiac nervous system. But what you can see is that there is high background, which is that blurred green. And the nerve signal is actually scrambled, what I like to say scrambled, because it's damaged by these clearing reagents. Now that couldn't have possibly been recognized because if you've never seen something intact, how would you know if you've messed it up? But in 2018, I started my PhD and I looked throughout the literature, and what I saw was just a variety of interpretations of the cardiac nervous system. And I said, there's no way our nervous system looks all different in the same types of mice hearts. There must be a processing defect here. And so I started my PhD and I said, well, I'd like to make a method that I feel confident in where we can clear the tissue and image it intact and look at the cardiac nervous system. And so that was my goal, was to use the best technology available at the time. Tissue clearing is just a technique that, I mean, there's dozens of papers published every year. And it was really readily evolving in the field. And I decided to take the approach called passive clarity. So first of all, tissue clearing is the process of removing pigment from an organ. Your pigment comes from lipids. You can remove them using heat and detergent. The detergent will bind from the lipids. The heat facilitates it getting out of the tissue. And in about 2 weeks, you will have a completely transparent organ. And as I mentioned, a lot of clearing techniques use very harsh reagents. The protocol I used, passive clarity, and I developed was able to, I optimized it to use the lowest settings, the lightest reagents, and really take a longer time in order to clear the tissue in the most gentle way possible. I knew those nerve axons were really susceptible to some of the harsher chemicals, and modified the technique and optimized the technique in order to have it maintain the axon architecture in the heart. I then also made this protocol compatible with immunostaining. Now, this is really an advancement in the field because at the time, this was that you could take your organ and you could have it labeled with a reporter endogenously, which I will mention, kind of break down that word in a minute, but you could have it reporter labeled, but you couldn't immunostain it. And so to break down the process of immunostaining, for those of you not familiar, if you have 3 cell types, 3 different types of cells in your body, I color coded them A, B, and C, you can target cell types to fluorescently label them with antibodies. This process looks like this. So cell type A will express protein A. Right now, you can't see anything. They're just cells in your body, but we need to be able to visualize them underneath a microscope. So cell type A, we know expresses protein A. We can use fluorescent antibodies that are targeted against protein A to what we call tag a cell. And so this antibody will specifically bind to protein A. Now we have some signal. Now we have something we can visually see under a microscope. And so we can take this under a microscope, shoot it with lasers, and what we're going to be able to see is a fluorescent cell now. And notice that only cell type A is going to fluoresce in green. It's because that antibody is specific. So you are able to target a cell type with a fluorescent antibody. So you'll hear me say, you know, I tagged the parasympathetic nerves with a red fluorescent antibody. What does that mean? I made the parasympathetic nerves fluoresce red. So For visual purposes, you just need to know that the parasympathetic nerves are red. You just need to know what color you're looking for. And I'll break down, what colors I'm using for everything. And not only was it compatible with one type of immunostaining, but of course you can also put other cell types into other colors. So I can also use a blue channel or a red channel and have multiple cell types targeted in the same tissue. Start looking at multiple different cells within the same tissue and through different processes. So the reporter line that I mentioned earlier. So rather than targeting with antibodies, you can also genetically modify animals or cells to naturally fluoresce a protein within your targeted cell type. So it's the same outcome. You're still going to see a color on your cell type. It's just two different methods of getting it there. And my staining and imaging and clearing protocol is compatible with both types. And this was really new in the field. And it's just in the last few years that more protocols have been able to be developed to be compatible both with immunostaining and reporter models. And so now I have a beautifully stained tissue. I can take it and stick it under a confocal microscope. For the purposes of my talk, you just need to know that confocal microscopy allows you to see fluorescence. It uses lasers to excite these fluorescence. And you need to know that it is giving me information about the 3D architecture of the heart. And so it's essentially, it's compiling all of the dimensions of the heart. It's able to see through the tissue and give me an output of 3D architecture. Now, when I look at the heart, it's quite large. It can be up to 22 days old of a mouse heart that's bigger than the size of a pea. We consider that a fairly large tissue to be sticking under a microscope. And so the issue that I came across was, well, if I want to image the entire thing intact, I'd have to do a really low resolution, a really high view of the heart. But I didn't want to lose that spatial quality. So what I did was some of the microscopes are calibrated for a picture stitching method. This is where you can take high magnification tiles and stitch them together. So these are 9 high magnification tiles, and then it's stitched together, similar to stitching together an image in Photoshop. And so then I'm able to get an entire view, high resolution of the intact cardiac nervous system. And so I was able to go back to the field and progress it one step further. Now, for the first time, seeing the intact, complete cardiac nervous system for the first time in up to adult mammalian hearts. And we were able to take this protocol and publish it in the Journal of Visual Education in 2020. And this really served for the basis of my thesis work. And so it's with this method development that I was able to go in and start asking questions about the cardiac nervous system. And the real goal of this project was to identify the 3D architecture and the remodeling that happens of the nerves during development, homeostasis, disease, and regeneration. For the purposes of today, I will be focusing on homeostasis and disease and regeneration. And this is a massive project, so I've divided this chapter up into 2 parts for today. So the first part really focused on looking at what is the nerve architecture of the mature heart, really setting the foundation for understanding proper and typical nerve architecture. And so I had to ask quite a few series of questions. So the way this talk will work is I will kind of lead you through some of the questions I asked and the answers, and then summarize what I found. So my first question was, going back to those 2 nervous systems, well, what's the distribution like between the parasympathetic and the sympathetic nervous system? To look at the parasympathetic nerves, we quickly found out it is not compatible with immunostaining. Actually, something that we've talked with others in the field, and they've had the same issue, and it's not well reported. And so instead, we took the approach of using a reporter model. So we naturally made the parasympathetic nerves fluoresce in red. We did this with a fluorescent protein, fun name, called TD tomato. So TD tomato will make all of your parasympathetic nerves fluoresce. For those of you interested in the genetics, it's quite a precise model. We have CHAT is choline acetyltransferase. It is a specific marker for the parasympathetic nerves, and CRE is knocked in under the endogenous CHAT locus. Cross it with a ROSA, lock, stop, locks, TD tomato, giving us our CHAT, TD tomato, parasympathetic nerve reporter mouse. And so I wanted to get the view of the 2 different nervous systems. So I took the hearts at what was considered a mature time point, postnatal day 7 or 7-day-old mice hearts. I will abbreviate postnatal day with P. And so postnatal day 7 hearts cleared the tissue and immunostained for the reporter protein, but also for the sympathetic nervous system. And so when I harvested these hearts and I stained them, you're really just working in vials and you can't see anything until you get under the scope. And when I took these hearts and I looked under the scope, I thought I would see a few nerve axons. I'll remind you that in the introduction, I said that the parasympathetic nervous system is thought to really have minimal to no contributions to the ventricular innervation. And so I thought I might be able to see a few new nerve axons. And so I looked for the red fluorescent parasympathetic nerve axons, and I was quite shocked because what I was able to see is that there were dense nerve axons covering both your right and left ventricles that I've outlined here. This was completely contradictory to all of the findings in the field at the time, and it really made us excited. And I know I came to Ahmed that day and I said something along the lines of, well, I have a thesis project. And so I was extremely excited. I can actually tell you under the scope, I was like shaking because I was so excited. And so I was able to see a dense network of parasympathetic nerves, but I didn't want to stop there. I wanted to say, okay, that's the parasympathetic nervous system, but what about the sympathetic nervous system? What does it look like in relationship? And quite a striking phenotype appeared where they looked nearly the same. And I remember being like, that's weird. Is this an issue? And it wasn't until I overlaid the two images and I took a very high magnification view, 100x view of these nerve bundles, and I was able to see that they're separate, they're just intertwined, and they're extremely close together. Now, most nervous systems might have regions of the parasympathetic and the sympathetic nerves being intertwined, but many regions where they're separated. All of the ventricles showed extremely close parasympathetic and sympathetic intertwinement. And so it was with this that we said for the first time, we were able to say that the parasympathetic nerves extensively contribute to the cardiac ventricles, and they are densely intertwined with the sympathetic nerves. Now, my other goal of this was to really set up what is proper nerve architecture. But when you start looking at these nerves and try to say, okay, how do you define normal nerves, right? It becomes really hard. You just have a lot of it kind of seems like random patterning, right? So I went back and I said, well, we often know that form follows function. So if there's a function, there's usually a form associated with it. And I said, well, what do we know the nerves function with? And I came to the coronary arteries. Your arteries respond to the nerves. They can constrict and relax. We know that's happening in the heart all of the time, right? To help with the pumping action. And so I said, well, we know that the nerves communicate with the arteries. Well, let's use this as a map, as a road map. We know very well in the field what the coronary arteries look like. We can cast them with dyes. They've been well studied in not only the mouse heart, but of course the human heart. And we know a lot about them. So what a good structure to kind of give me a roadmap of proper innervation. And so in order to look at this, I used a different reporter model. This time I made all of the arteries fluoresce red. And I combined this looking with all of your nerves, this time in green. And for those of you interested in the genetics, I used a Connexin 40 Cree-ER mouse, and it's crossed again with the Rosa lock-stop lox TD tomato, leading to Connexin 40 TD tomato. Again, I made all of the arteries fluoresce red. And so when I first looked at the heart, I could see, okay, my arteries are fluorescing red as expected. You've got some beautiful, your left and right coronary artery. Here I'm showing you a lower portion of one of your larger left coronary arteries. And then I stained it for all of your nerves. So this is just all nerves, parasympathetic and sympathetic and even sensory. And we could see, yep, it's stained for nerves. We see them there, but it wasn't until I overlaid the two images that I could really see what was happening. These nerves were really closely associated and aligning with the coronary arteries. And when I took a high magnification view, this really came into a clear picture of the fact that there's a nerve axon and there are these beautiful nerve projections leading into the coronary arteries. This is what's allowing for that regulation of vasoconstriction, vasodilation of artery dynamics. And it's really only in 3D that you could get a view like this. And so, like I told you in the beginning, all of these images are in 3D. And I have been very lucky to have two amazing collaborators, and they have taken all of my work and been able to take a largely qualitative project and bring quantitative measures into it. So we modeled all of these systems in 3D using Amaris 3D modeling. Now, this branch was led by Porva Halbe, a master's student in our lab, where she accurately and precisely and painstakingly took the time to trace out all of my nerves and all of my arteries and all of my hearts throughout embryonic development and mature development. And it begins to look something like this. She uploads the heart. Porva is able to trace the nerves in green, arteries in red, and we can see in 3D how closely the nerves are associating with the arteries. And they're really aligning quite strikingly and innervating quite strikingly the coronary arteries. And this is the first time that we've been able to model the neurovascular system of the mature heart. Many times people have looked at the arteries, people have attempted to look at the nerves. This is the first time we've been able to put it all together. And like I said, this is quantitative. We have done extensive quantification throughout embryonic development, and that was also with a ton of help by William Kasberg, a graduate student in John Audia's lab, and he was really our Amaris expert, helping to make sure that we were doing the correct statistical analysis, making sure that we had a good pipeline for analyzing all of this. And I won't get to highlight just the extensive amount of work we've done through embryonic development, but I can show you that we've been able to quantify a ton of phenotypes. I'll highlight here that we were able to define about a 35% nerve artery association in that anterior wall. We've, of course, looked at both sides of the hearts. We've also looked at the veins, but we were able to really define a normal nerve architecture being, around 30% nerve artery association in the heart. And so to summarize these two parts, I've shown you that the parasympathetic and the sympathetic nerves are densely intertwined in the ventricles. And I've also shown you that the nerves align and innervate the coronary arteries. And this served as a basis for me then to move on to part two and a really fun part of this project, where we said, okay, well, let's use this normal architecture to understand how nerves are remodeled under a disease and regenerative setting. And this required, again, a series of a few different questions. We were very focused on understanding this because the leading cause of death, not just nationwide, but worldwide, is cardiovascular diseases. Coronary artery disease will cause about 1 in 4 deaths, or 18 million deaths annually. It is unmatched. This is because there are no curative therapeutics. We can try to prevent it. We can try to put a stent in the heart. But if you have an injury, it's really an irreversible condition. And so it's an unmet need that we need new therapies to help with coronary artery disease. It's causing 30% of deaths worldwide. So what is coronary artery disease? Typically, you have a lovely, healthy coronary artery. It's flowing blood, no resistance, but due to diet and other genetic factors, you can get the buildup of plaque. Plaque can eventually block blood flow. And that is where you can have a myocardial infarction, in clinical terms, better known as a heart attack. So I'll ask that you remember that MI means either heart attack or myocardial infarction, because I'll mention MI and I'll have MI abbreviated quite a bit. So MI is standing for heart attack or myocardial infarction. So what happens when a myocardial infarction happens? The blood flow is blocked, that whole entire downstream tissue dies. About 1 billion cardiomyocytes will die. The heart can regenerate about 1% of cardiomyocytes a year. So that is completely insufficient. The heart has to compensate in a different way. It just lost a ton of tissue. And so instead, it's going to develop a non-contractile scar. I've shown that here in gray. It's this very thin scar tissue, and it cannot contract. It cannot help your heart beat. It is just patching the wall. Now, if I take a section of this heart, if I slice this heart in half and show you how severe this injury is, it becomes very clear how much tissue you lose. So in a healthy heart, you have this beautiful red, healthy myocardium. After an MI, this blue is scar tissue. So it's extremely thin, and it'll even fold in on itself. That's how weak it is. And that's what's replacing that nice, healthy, strong tissue. So you can imagine that, the pathology of this disease is extremely severe, and that's what's really contributing to that high mortality rate. But, we focus a lot on the loss of tissue, but that's not what people are dying from. They're dying because of the nerve damage. So you have these beautiful, healthy nerve axons in the diseased heart. What we're going to see is regions of hyper or increased innervation, as well as regions of denervation. And it's between these two that we're going to see that the nerves are going to cause misfirings. Those misfirings cause arrhythmias. Those arrhythmias cause sudden cardiac death and heart failure. And so we're seeing that the nerves are a really key pathology to causing sudden cardiac death. And actually, denervation is a direct predictor of the likelihood that you are going to have sudden cardiac death. So the more nerves that die, the more likelihood that your heart can stop beating after an MI. And so when we think about this condition, we think of it as an irreversible process. You have a healthy heart tissue, and you will be forever stuck with today's medicines with a diseased heart after that. So how do we start thinking about how to reverse this process? This is where regenerative medicine comes in. What processes can we tap into to tell this diseased heart to repair itself? How can we get the heart back and functioning again so we can therapeutically help people post-MI? And when we think about trying to identify new regenerative processes, we think about trying to model something that could show us what to look at. And that's where our model comes in of the neonatal or the newborn mouse. Newborn mice, when I'm referencing these are 1 day old mice, and these are models for heart regeneration. So what does that mean? So a neonatal mouse is about the size of the top of your pinky. And if given a similar injury, so our lab specializes in an injury technique where typically you have plaque that's blocking that blood flow, we can surgically tie off that blood flow with a suture. And so we are taking a 1 day old mouse, again, size of your pinky, are looking at its heart, which is smaller than the size of a pea, looking at its artery, which you can imagine how tiny that is, and tying a knot around it. And that was Ahmed's idea. And it worked. It worked quite strikingly because when we looked 21 days later, there were no defects of injury. There was no scar tissue. There was completely new tissue. No defects in contraction. It completely regenerated. So now we have a model for heart regeneration. We have a process that is happening naturally in an organism that we can start learning about. But what do we contrast it to? Well, we go back to the mouse because this regenerative response of the newborn mouse shuts off just 1 week after birth. They can only repair its heart for 1 week after birth. You take the same mouse 7 days later, 7 day old mouse, perform the same exact surgery, it's going to respond much like a human heart. Scar tissue, pathological nerve remodeling, and ultimately arrhythmias. And I was very curious about the nerve responses. Ahmed mentioned we have previously been able to show that heart regeneration is dependent on nerves. If you inhibit the nerves, the heart can't regenerate. And this led way to me really questioning. So I looked in the literature, I saw, okay, that neonates, they contract normally, they don't have arrhythmias. And we know it's a nerve dependent process. Are the nerves, like, what's the plasticity of the nerves? How are the nerves responding under this setting? If we look at a 7 day old mouse, we know, and there's much data about this, that the nerves are undergoing denervation, heterogeneous remodeling, very similar to what I showed you in the adult human heart. And so I really wanted to understand, what is this process? What is the differences between these two processes, the non-regenerative and the regenerative heart in respect to the nerves? And so I really wanted to first look into that nerve plasticity. I wanted to watch the nerves. I said, I have all the techniques to look at the nerves in 3D. What happens if I watch the nerves in the regenerated heart over time? And so 3 weeks later, the heart will be fully regenerated. And I looked at it over this time course. So I performed a myocardial infarction surgery in either the P1 or the 1 day old regenerative mouse or the non-regenerative P7 mouse. I looked at their hearts at 7, 14, and 21 days, really trying to get a scope of how the nerves were changing over time. And before I show you the data, it's important that you know 3 different regions that I looked at in the heart. The infarction zone is the region directly downstream of where I tied off their coronary artery. This is where the tissue in the non-regenerative model is dying. It's directly impacted by that injury. So you could think of it also as the injury zone, infarction zone, injury zone. They're both going to say IZ. Think of it however you'd like. Border zone is the region next to it. That's where you see a lot of nerve pathology in the non-regenerative heart. And then the remote zone's way of internal control. I thought, oh yeah, it's the remote zone. It's not injured. This will be a good kind of marker is one of my controls in this experiment. I also wanted to look at an uninjured heart just to start. So I took the entire uninjured heart and I looked at these 3 different regions. They all have nerves. That's good. That's expected. I didn't injure this heart, so I'd hope that they're innervated. And so I saw remote zone, border zone, and infarct zone, area away from the tissue or injury, area beside the injury, and area in the injury zone, and saw nerves. So that's my kind of control I'll allow you to compare to through these images. And then I looked at the regenerating heart first. So regenerating heart, I looked at 7 days post-injury when it's just beginning to regenerate. What I was able to see is a striking phenotype. I was able to see that the remote zone looked relatively normal, but the border zone and the infarct zone both became denervated. I saw a complete lack of nerves in both areas with, some little nerve fragments in the border zone, but pretty much completely denervated. Just 1 week later, 14 days, I saw a really nice progression where I then saw remote zone again, looked pretty normal. Border zone began to be re-innervated, and the infarct zone even showed some new nerve axons. And at 21 days, when the heart is fully regenerated, I saw a progression where both the remote zone, border zone, and infarct zone was completely re-innervated. And if you compare fully regenerated heart to your control, it has nearly restored perfect levels. But this is just by eye. We can quantify this again. So I started by 7 days post-injury. This graph is showing you the percent of coverage by nerve. So how much area are the nerves covering? And the gray bars are your controls. So those are age-matched control samples that are not injured. So I said, okay, in comparison to non-injured hearts, what do these regions look like? A new phenotype popped up. I was able to see that remote zone didn't actually look as normal and uninjured as I thought. There was a compensatory increase in the remote zone innervation, and then the border zone and infarct zone was still decreased. 14 days post-injury, I saw still that increase in remote zone innervation. That was really interesting to me, seeming like the remote zone was able to bring in some new nerve axons to increase innervation now in the border zone, but still decreased in the infarct zone. And 21 days post-surgery, really strikingly, all the levels were restored. Remote zone was no longer hyperinnervated or increased. It was back to normal levels. Same with border, same with infarct zone. So we could say that the axons are extending from the remote zone to re-innervate that border and infarction zone during neonatal heart regeneration. I also wanted to compare this to a pathological heart, non-regenerated heart. So performing a 7-day-old MI surgery and then 21 days post-injury, and just to kind of highlight the pathology here. Pretty normal remote zone, but border zone, we're seeing a lot of heterogeneity. We're seeing partial denervation, hyperinnervation regions. And then the infarction zone, we're seeing complete denervation. So this is contributing to the pathology of a non-regenerative heart. We can quantify this now. The pathology, the diseased heart is in the striped bars, and we see the remote zone is normal, but we've got a ton of heterogeneity in the border zone. Some hearts super high, some hearts super low, and the infarct zone is just strikingly denervated. So we are able to confirm, and this really supports findings in the field, that we're going to see the pathology in the border zone with striking denervation in the infarction zone. I next wanted to go back to some of my main phenotypes that I was able to show before, and I wanted to ask, well, we see that the nerves are there, but I mean, are they normal? That wasn't enough to convince me that they were normal. So I wanted to look at their nerve architecture, and so I wanted to look into the 2 nerve subtypes, the parasympathetic and the sympathetic nerve architecture. And so to do this, similar setup, an MI and the regenerative or non-regenerative heart looked at 21 days post-injury when either regeneration is complete or when the pathology set in, cleared the tissue and went back to that CHAT-T, tomato tyrosine hydroxylase, that comparison between parasympathetic and sympathetic nerves. And what I was able to show was in the diseased heart, here we have the blue is where I tied the suture, the white is the infarction zone or the area that was directly injured. And I was able to show that in the border zone, we again saw pathology, but for the first time since I had both nervous systems here, I could see a new phenotype. We saw that it was only the sympathetic nerves that were sprouting. Now, this is in line with literature. There's many literature that says, okay, your arrhythmias are getting generated by your sympathetic nerves sprouting. But what I could show for the first time was the architecture of the parasympathetic nervous system, which didn't sprout. And for the first time, I was able to also see disassociation between the parasympathetic and the sympathetic nervous system. I also looked into the infarction zone and saw that it was completely denervated, similar to my other phenotypes. And when I looked into the regenerated heart, it was quite a striking comparison. I was able to see dense axon bundles, and strikingly, those parasympathetic and sympathetic nerves were densely intertwined again. This was the same for the infarction zone, where the entire infarction zone was re-innervated by both parasympathetic and sympathetic nerves, and that these were again bundled together. I didn't want to stop there. I wanted to still look into the architecture of the regenerated heart, and I wanted to look back into the arteries. Luckily for me, Christy Redhorse has done some beautiful work looking at how the arteries are remodeled in the regenerated heart. And she's been able to show a very unique process that only happens during neonatal heart regeneration. So what happens in a control heart is you have your right coronary artery and your left coronary artery. They are not coupled. We see that they are completely distinct. Their branches will not bridge. But if given a myocardial infarction or heart attack, we see that the neonatal mouse, by 4 days, it's an extremely rapid response, 4 days post-injury, will build a bridge to bridge the right and left collateral or coronary artery. This bridge is called a collateral artery. It's allowing for, in control hearts, you see they're separated. 0 days or 0 hours after infarction, they're separated. 4 days after injury, it builds a bridge, allows for the restoration of blood flow, and allows for complete regeneration to occur in that mouse heart. And so I knew that the collateral arteries form, and this created a great model for me to say, well, there are now arteries, are there nerves? And so I wanted to look at if the collateral arteries showed nerve association. I looked at 7 and 21 days post-injury in the regenerated heart, cleared the tissue, and went back to using the Connexin 40 arterial reporter. So I had all the arteries fluorescing red and all of the nerves in green. And when I did at 7 days post-injury, I thought I might be able to see some nerves, but again, I was able to see that it was pretty strikingly denervated. I'm showing you just high magnification views of an infarction zone. So just that injured region. So there were no nerves, and that actually matches, what I had seen before, that infarction zone is pretty denervated, but the collateral arteries were built. We see this nice artery bridge. 21 days post-injury, I could see that the nerves and arteries were able to regain association really nicely. But this is just telling me that the nerves are near the arteries. It doesn't tell me that this was recruited due to any dependence or any mechanism. So I wanted to take this again a step further and say, do the nerves require the arteries to be formed to reinnervate the heart? Is this a required relationship where one depends on the other? Or is this just happenstance, the nerves are near the arteries? And so in order to do this, I used a genetic model. And really, for those of you in the audience who are not familiar with a Cree and a Floxed model, the only thing you need to know is that I stopped the formation of collateral arteries. I stopped them. I inhibited them from happening. For those of you interested in the genetics, I used that arterial cell Cree and crossed it with a CXCR4 Floxed mice. CXCR4, CXCL12 ligand receptor pair allows for arterial cell migration. If you inhibit one or the other, the arteries can't form. And this has been published by Christy Redhorse's lab. And so we use their model to our advantage, saying, if we inhibit collateral arteries, do we see nerves? If there are no nerves, the nerves need the arteries. If there are nerves, nerves don't need the arteries. What I was able to show in the control heart is that it's completely reinnervated. In the inhibited heart, there are no nerves. This is telling me that the nerves are dependent on collateral arteries to reinnervate the heart. And it's with these main findings, the restored levels of innervation, the bundling of parasympathetic nerves, the nerve association with the collateral arteries, and the dependence on collateral artery formation, that we were able to define a new phenotype of neonatal heart regeneration, which is physiological reinnervation during neonatal heart regeneration for the first time. And so this really leads way, and circling back, that nerves could be a great therapeutic approach, that nerves are able to show quite a level of plasticity. We just need to start understanding how, because if we can tap into how to get these nerves to reinnervate the tissues, maybe we can stop those arrhythmias and those sudden cardiac deaths. that are happening after MI and causing heart failure. So the nerves are a very promising therapeutic for heart failure. And so just to very briefly follow up, there are so many questions that came from this research, and I won't highlight them all because you'd be here all day. But one of my favorite questions are, how are the nerves recruited during regeneration? Because if we're going to use this as a therapeutic approach, we need a target. We need to know what is telling the nerves to come into this tissue. And so I showed you a timeline where the nerves are able to get recruited into the regenerating myocardium, being denervated, partially reinnervated, and fully reinnervated. And luckily, there is a wealth of information in the field about nerve development in the heart, and we can start tapping into what we know about development and asking if those questions are also active during regeneration. We often see an overlap between development and regeneration because they're both growing a new tissue, just in different ways. And so one approach that I've highlighted is single cell transcriptomics, which basically it's looking at different cell types and asking what they're releasing or secreting. And so some cell types that I've highlighted is vascular smooth muscle cells, endothelial cells, fibroblasts, and cardiomyocytes, and some factors that we know recruit nerves. I really highlighted a lot in my thesis neuronal growth factor because it is involved in nerve development. And I thought this is a great factor potentially to target as a therapeutic. And it's also a factor that's been associated with pathology. And I'd love to talk more about that if anyone would like to later. And I would like to just highlight that I couldn't share all of my work today, but I was able to also have some wonderful collaborations while I've been here. I also worked on a project which focused on a protein leucine-rich repeat-containing 10, LRC10, in collaboration with Young-Suk Lee and Tim Camp. And very exciting news that yesterday we received information that it has officially been accepted under Nature Regenerative Medicine. So that will be out shortly. And I also worked on a wonderful project with, at the time, a postdoc in the lab, Jiyoung Bae, who is now an amazing PI at Oklahoma State University on her project, which looked to leverage the metabolism to stimulate adult cardiac regeneration. And that paper was published in Circulation in 2021. Also, my nerve paper is currently under review in iScience, and yeah, maybe next week I'll get it out. And it's with that, I will pause before doing acknowledgements. I'll do questions first, and thank you for your attention. We have these fun boxes. It will be on in a second. Yeah. You ready?
Rebecca, that was so beautiful.
Thank you.
Thoughts in your introduction that the sympathetic nerves innervate the cardiomyocytes and they control a lot of functions. And then you showed really beautifully that the parasympathetic nerves innervate the coronary arteries. But do you think the parasympathetic nerves also innervate cardiomyocytes?
Yes.
And what would that role be in development and regeneration?
Oh, what a wonderful question. I completely think that they innervate cardiomyocytes. The level of sympathetic innervation, it's speculated that every cardiomyocyte in your heart touches a sympathetic nerve. That's an insane amount of innervation. We're seeing that there's a similar amount of innervation of parasympathetic nerves. It would be mind-blowing if they couldn't touch a cardiomyocyte. I don't even know how you dodge a cardiomyocyte in the heart. And I think they have tons of regulatory processes in the cardiomyocyte. One of my favorite ideas is an idea that maybe there needs to be a balance between both of them. Possibly, does every cardiomyocyte need either a parasympathetic or sympathetic nerve? I'd like to believe that they need both in order to have a balance. And that when we see nerve pathology, like I showed, only sympathetic nerves are sprouting in a pathological model, maybe that's throwing off then that balance. And so I'd like to think that they're really regulating a lot of the same functions, but potentially providing a balance cue for the sympathetic nerve so you don't have too much activity. Because, you know, when you look at the heart, it's all about a balance. It's a striking balance, yin yang of the two nervous systems. And so I would like to think that it's influencing. We've seen proliferation, which was also shown in sympathetic nerves. I'd love to start looking into data over cardiomyocyte size. because that's been really a striking phenotype of the sympathetic nerves. Oh,
Do I have to press it?
yeah, you speak into it.
Oh. Wonderful talk, Becky.
Thanks, Oz.
I was wondering if I could pick your mind about the 7-day post-MI and the 14-day post-MI in the remote zone increase in innervation, maybe a reasoning behind that.
Yeah, that was a really, I didn't expect that. I will say I quantified it and I was like, oh, that's cool. And it's possible that the nerves are becoming denervated, but before they can go in to their new zone, while that zone is turning over cells and regenerating, that they're kind of building up. And then they're, floodgates are released and then they're there and they can re-innervate. So I think it's a temporary buildup, a block up, as they're kind of prepping and waiting until the tissue is, of good standing to go into the tissue. One interesting idea, that I've seen in the regenerated heart is it seems that the nerves kind of wait. They're very delayed. A lot of things we see increased within the week after regeneration, but we see that the nerves at a week look terrible. And so it seems like they wait until the tissue's in a better standing, more restored state before re-innervating. So it seems like the nerves want to come in last. Interestingly, in embryonic development, the nerves come in really late. The mouse is born at 21 days after embryonic gestation. The nerves come in around E15.5, 16.5 after the tissue's pretty well established. So potentially the same thing, it's waiting for the tissue to really be in a good standing before going in, or maybe that might cause pathology if they come in too soon. So maybe that's kind of the buildup before it goes. Yeah.
Awesome, thank you.
Yeah. Awesome. Oh, there's one in the back. Yeah.
Most of the neurons in the heart, I think they're called up to the top of the heart.
Mhm.
Do you have any sense whether it's the infarction that happened in that area that
Yes. 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 So the question was the fact that there is the nerve somas are located at the top of the heart. Does the injury directly injure the nerve somas, or how is that process happening that we see these axons re-innervating the heart? I use the term re-innervation a lot. I could also have said axon extension. So the nerve somas seem to be fine, but I will say there's tons of pathology in the nerve somas where we see remodeling of the nerve somas during a pathological state. We can even see that they kind of become mixed up in what their identity is, where they're kind of sometimes an intermediate between parasympathetic and sympathetic nerves. So one of the ideas in the field is that they become like able to express both features of both parasympathetic and sympathetic nerves. So we've seen defects in the somas for sure. But as far as we know during the process of neonatal heart regeneration, it just seems to be the axons that are responding. And that's pretty common in your peripheral nervous system, that it's really your axons that are doing the heavy lifting, the work of regenerating. And it's, your somas are very well protected. So there are somas directly in your heart as well as in your spinal cord that are both contributing to the parasympathetic and sympathetic axons of your heart. Excellent. Yes. Yeah.
Do you have any further insight into what's going on in the border zone where you see that sort of heterogeneous?
Yeah.
Is it some is it a continuum or is it sort of two distinct populations?
Oh, yes. It seems to be two distinct populations from our understanding. Seems to be two distinct populations. It also seems like the sympathetic nerves can show a lot of pathology just all over the body. So you can actually see overall, if you have a heart attack, your entire body will increase its sympathetic tone. So it seems to be a huge remodeling of your entire sympathetic nervous system. And that response is really causing a lot of the pathology in the nerve axons, but also just overall in your body. So that's why beta blockers are a huge treatment. That's going to stop or lower, not stop, but lower your sympathetic activity. And so those have been very successful in helping against arrhythmias. But you're seeing it not just in the heart, but also in the entire body. And then did that answer your question? In the border zone. Yeah. The hyper or the heterogeneity. So what about the heterogeneity in the border zone? Well, that's also been related to growth factors. And I have an idea that, again, kind of going back to what we see in the regenerated heart, is the heart needs to be pretty established in its tissue structure. And I think it can't go into that scar zone too far. And so it's kind of trying to reinnervate the heart, but it doesn't have good targets. And since it doesn't have a good target, we know it needs to communicate with cardiomyocytes. We already lost all its cardiomyocytes. And this connection between maybe nerves and cardiac fibroblasts, or potentially an axis that's really causing a lot of pathology. And then just, yeah, the lack of targets, I think, could be a really good understanding of why that border zone might be causing a lot of the pathology. Because the border zone is injured. There is quite a loss of tissue that's right on that periphery of between healthy cardiomyocytes and fibroblasts. So, and the cardiomyocytes are just also out of sync, right? They're not electrically coupled. So if they are coupling with cardiomyocytes, they're not coupled correctly to the entire conduction system. So that's what I would speculate. Excellent. Thank you. I'll take one more question just so we can. Yes. Yes. Oh, this is very interesting. Yeah, so it's a very interesting dynamic where I'm seeing that. And I mentioned that the nerves can regulate proliferation. Really interestingly, the sympathetic nerves are a negative regulator of proliferation. So it would behoove the tissue to not have a signal that says stop growing in there. And so one of my hypotheses is that the nerves signal for, and we see also in different contexts that nerve signaling can vary to either tell the tissue to grow or tell the tissue to stop growing. And then at homeostasis, we see it's maintaining actually a balance of growth. So my guess is that it can be also a negative regulator of proliferation, which we have evidence for. It can lead to cell cycle exit, circadian rhythm genes. And so my idea is that the nerves can function as a negative regulator of proliferation, and we don't want them there yet. Also, another idea is that for the nerves to really reinnervate the tissues, those tissue types need to be solid. You can imagine renewing a cardiomyocyte and trying to innervate it at the same time is a very tricky process versus the cardiomyocyte being, leveled off and established, and that ECM or extracellular matrix is more solidified, that then the nerves come in. So that's what I'm thinking. Okay, and just so I get out here a little bit on time, I would love to go into acknowledgements. I have so many people I'd like to thank. First, I'll start with my mentor, Ahmed. Ahmed really has just allowed me to be me in the lab. Come in with my enthusiasm, and he pitched a project to me right when we met, and I remember thinking, this is the coolest project I could have asked for, and I'm telling you 5 years later that it is the coolest project I could have asked for. During this project, most of the time stuff was not working, and he was very quick to just shrug it off and say c'est la vie, which means such is life, and it just allowed me to continue on, shake it off, and keep going with my experiments and not really worry about it. It never fazed him that things weren't working. I would freak out, and it was like, yeah, it's fine. It's science. And I needed that balance. And it just, I never left his office without ideas. And so I always was able to drive back into the lab, enthused, excited, and really never deterred that this can work out. And I think we always, had like little breadcrumbs that were working that always kept me along. And I'm so appreciative, Ahmed, for all of your patience and kindness and support over the last 5 years. I still think this is the coolest thesis project I could have ever dreamed up and ever asked for. And I'm insanely proud. And I'm really thankful for the independence you've allowed me. I really needed it and wanted it and desired it. And you really just allowed me to run with my ideas, even if they didn't work or weren't good. But the ones that did work worked pretty well. So anyway, you really, excited me about science, and it's one of the major reasons why I'm going to stay in academia as of now and continue on for a postdoc. And I'm very excited to see what I can find next. I'd also love to highlight again the people who've helped directly with the NERV paper. Porva has just been so phenomenal. She's a master's student. She's put so much time into analyzing all of my data, allowing for this project. I think it was my second year when I came to you and was like, what if we modeled the nerves in 3D? I never knew this was a thing. It's a newer software. And it wasn't until last year that I could find someone, we'll say courageous enough to join. And that was Porva. And man, nothing deterred her. She was just like, yeah, let's do it. You don't know how to use software? Sounds great. Let's do it. Let's try it. And she was able to work alongside William, who knew the software and could give us a couple pointers of where to go. And it's between them two that I honestly think it's the most successful collaboration, where I could just pass off images, crank out images. They were quantifying, and it just took this paper to a whole nother level. So I'm so thankful for both of you, just your ambitious and joining into my work. I'd also love to give a huge shout out to Jiyoung Bae. Everyone who knows me knows I love Jiyoung. I got to work with Jiyoung for 4 out of my 5 years. And honestly, it's one of the luckiest things that happened to me in the lab. Jiyoung is the best person to work beside. She is a phenomenal scientist. She's the most talented scientist I've ever gotten to work beside. She has just such a rigor, such a passion, such a level of excellence. And she told me very early on, I don't repeat things. And I have tried to approach science with the same mindset of, I'm going to try to do the experiments to the best of my ability, where I don't repeat at least the same mistake twice. Jiyoung tends to get it to work much faster. And Jiyoung, it's just been an amazing pleasure to have worked beside you, and then just also had you not only as a mentor, but a great friend, and just taught me how to be a great scientist and a great human as well. I'd like to also thank the entire Mahmoud Lab for making my experience so enjoyable. Timothy and Wyatt have spent the most time with me in the trenches, and I really appreciate them making it a good time being there. And then Dakota has just joined, and it's been great having him in the lab, as well as Alyssa being in the lab. So I'm really appreciative for such a supportive crew to be surrounded by. I'd also love to highlight our collaborators. Like I mentioned, I was able to work with Tim Camp and Young Suk Lee on a side project, and I learned so much from them and how they ask questions. And it just became a really fun side project that really ended up paying off. Also, a huge thank you to my committee members. Grace, Rupa, Aki, and Francisco have provided so much support and feedback and kindness throughout my time here. And I really appreciate their thoughtfulness. Of course, Grace, you've been just phenomenal. You've always given me that oomph that I needed. That's that encouragement that I knew I could just keep going, and just those little nudges that I needed of you're going to make it. You're going to make it. You're going to do it. I think I might make it. And I've also had some great core facilities that have supported me wonderfully through my work. The BRMS core, our mice line was run by Conrad. He just did a phenomenal job of jumping in and helping with my transgenics, so I could just really start cranking out some other data. Optical imaging core Lance is just phenomenal in keeping these scopes up and running and allowing me to, again, just show up and do my thing without having to think about it. So that support was invaluable for my thesis work. I'd also love to highlight the genetics training program. I do brag that the genetics program is the best. And it's because of really, they just solicit feedback from students. They want to do what's best for students. And I will say, not one time did I ever feel like there was a lag in the program asking, what can we do to be better to students, and allowing students to serve on committees, give feedback. And I'm so appreciative for Nicole, Francisco, Martha, and Audrey for all of your support. And I know us students are always, we're asking for something, but you guys do just honestly a wonderful job of keeping the genetics program moving forward. And I'm so thankful for it. The Department of Cell and Regenerative Biology, CRB, was honestly a great community to be a part of. I came from a small liberal arts, honestly terrified that I was going to such a giant institution, but I quickly landed in CRB, and I am so thankful for the people within it. Deneen, Barack, Valentina, and Colleen have all been so supportive and really making me feel like I'm still at a small liberal arts. And I really appreciated it. It took all the elements that I had at Fort Lewis and brought it to UW-Madison. And I do really thank my experience at Fort Lewis for really providing me a foundation to pursue graduate school. And a huge shout out to my past mentors, Katie Mouzakis and Steve Fenster, both from Fort Lewis, and Sanjay Jain from WashU in St. Louis. Without them, I couldn't have really had the skill set or the mindset or even the desire to pursue research. And a huge shout out to Katie Mouzakis. She was my first research mentor, worked under her for 2 years. And she showed me how to be enthusiastic about science, how to be independent, and was so thoughtful with her mentorship. She really took the time to make sure that I had a solid foundation in understanding how to pursue independent research, and I'm really thankful for that. I'd also like to mention just the great help that therapy's been, and my therapist, Sarah, has really just been phenomenal in providing me the support to support my mental health and just continue on. And, you know, through those tougher times of graduate school, it's not a linear path. It's a pretty tough journey to get through. And I'm really thankful to have had that support system. I'd like to also just thank my friends. I have the best friends in the world. And anytime I just like need to be thankful, I think about my friends and my family, because I'm incredibly thankful and surrounded by just people I aspire to be like and make me better. Ashley, I've known since high school. Becca and Kelly, I've known since undergrad. We all were in graduate school at the same time. Megan, Sierra, Muhong have been here with me at UW-Madison, and they have made my time here, like, those are my memories of UW-Madison, or my times with my friends. And we made plans to see each other. A little bit after the pandemic, we all went to Zion, Becca and Kelly. That was really fun. Ashley got married. I mean, it's just a ton of fun. We spent a lot of hours at the library meeting up, and I'm really thankful to have just friends that I aspire to be like. I'd also love to give a huge shout out to my family. My mom, my dad, Stuart, Andrew, and Matt. I have 3 brothers and a twin brother. And everyone has been incredibly supportive in family vacations and family get-togethers, and listening to me science ramble, and then just being like, I don't know what you're talking about. Please tell me you know what I'm talking about now. Thank you especially to my mom. She's just been wonderful. She's always been the phone call I could make to get cheered up. #1 support. And thank you to my brother and sister-in-law, Todd and Amanda. We went every year to go visit them, and it's something I always looked forward to. And then a huge shout out to my grandparents. My grandparents are the greatest people I could imagine. And my grandpa unfortunately passed away a couple of years ago, but they were always the FaceTime call that I needed. My grandma will watch this later on Zoom, so hi, Grandma. And I could always just count on a FaceTime to brighten my day. We spent a lot of time in the kitchen together. They really instilled my love in baking. And when I came to graduate school, baking was really always my escape from lab and what I could do to really relax. And I always thought about them. And then when, I'd bring treats in, I got the comments a lot, well, a great baker makes a great scientist, because following a recipe is so similar to following a protocol. And if something you have to substitute something in a recipe, well, you have to substitute a reagent in a method. And I can't imagine developing, my own method without the lessons that they taught me in the kitchen, and really just their constant support of me. My grandparents would ask me, what's your major again? Say it slower. So they could write it down and tell like the grocery store clerk. And it's with that I've dedicated my thesis work to my grandparents. So I love you. And last but not least, his own slide. My lovely fiance, Oz. Oz has been the #1 support system that I could have asked for. We have been together for nearly 8 years. He came, pulled him away from Colorado, pulled him away, said, you want to go to Wisconsin? And he didn't bat an eye. He came right on over, and not as pretty. Um, but we made the most of it. You know, Oz just made my whole experience, and he makes my whole life just so enjoyable. I love every day with you. You've allowed me to really, especially in the last 6 months, just crank out data while I do nothing around the house. And he has done everything, and he makes life so fun. I'm so excited to see where we go next, and just our journey of life. Every day with you is the best that I could ask for. And I'm so thankful to have such a supportive, wonderful, kind, loving, handsome partner. And our cats, Bunny and Clyde, would be a miss. We love them. And with that, I would like to just thank you all for being part of my journey. Yay. Yay. I'll turn the lights back on. Thanks.
I feel like I learned a lot. I was like, holy cow, I learned too much.